video we are going to understand the overview of blotting techniques. So there are different type of blotting techniques, but in this video we are going to look about the common steps and what is blotting. So first, what is blotting is the process of transfer of DNA or RNA or protein from a gel to a membrane is called blotting. So we separate our DNA or protein or RNA in a gel. So from the gel we are transferring our sample to a membrane. So this process is called blotting in simple terms. Overview. So in common blotting techniques there is a common 5 steps. So what are the common 5 steps? Are first is gel electrophoresis. So we will be separating our sample DNA or protein, we will be separating in a gel. Second is the blotting. The sample is transferred from the gel to a membrane. So it's a nitrocellulose membrane or nylon membrane or PVDF, polyvinylidene fluoride. Third step is blocking agents. We add blocking agents on the membrane to prevent a non-specific binding. Fourth is hybridization. So we add probes to specifically select a target sample or DNA or protein. And fifth is visualization. So we visualize the selected target protein or DNA with autoradiography or chemiluminescence technique. So now you might think why we are doing all these things. What's the reason to do blotting? So if we want to analyze a specific DNA or specific protein, we can't be just picking it from the gel, because we separated based on their size, and from the gel we can't be taking the DNA or analyzing it. So we transfer it into another one membrane. In that membrane we can perform on our analysis. So whether our DNA is present or whether the protein is present. So these kind of things we can check in the membrane. We can't do it on the gel. So that's the reason we are performing this blotting technique. So let's check what are the blotting techniques are. 4 important types of blotting techniques are northern blotting, southern blotting, western blotting, and eastern blotting. So northern blotting is to analyze RNA. Many people used to confuse which is to analyze which sample. So they might confuse that southern blotting is RNA or western blotting is DNA or northern blotting is protein. So there will be some confusions for students. So to remember it easily, northern blotting. So in that north, R is there. So R for RNA. That's all. You just fix that thing, and everything others you can write it easily. So northern blotting is for RNA. It is discovered by James Alvin, David Kemp, and George Stark. Oh, Stark, Einman, I think. So in 1977 they discovered this technique. Coming to southern blotting, actually this is the first discovered technique. It is discovered in 1975 by Sir Edwin Southern. So from this only it has been started. So southern blotting is to analyze DNA. Okay, because of his name they named it southern blotting, and when discovering blotting of RNA they named as northern blotting, just as a complimentary. And coming to the western blotting, it is discovered by Harry Taubin in 1979. So this is to analyze protein. So like southern is for DNA, and you can, another hack is you can remember D for down. So down means south. So southern is for DNA, down. Okay, and northern blotting is for RNA, north, and western blotting is for protein. Eastern blotting is hardly used, and it is used to analyze post-translational modification. So the protein after translation, there will be some acetylation or histylation or methylation. This kind of modification is there. So to identify those things, we use the Eastern blotting. So PTM means post-translational modification. So these are the important 4 types of blotting. Apart from this 4, there are other blotting techniques also. Like there is reverse northern blotting, which we use RNA to detect DNA. So DNA is detected using RNA, and there are far western blotting. So in that we will study protein-protein interaction, and a far eastern blotting. In that we will study glycolipids, and there is dot blot, which means a concentration of protein is detected. So these kind of other blotting techniques are also there. So in this video, we check the overview of blotting technique, how it is done, the common steps in all the blotting techniques, and what are the types of blotting techniques, why it is used for. So there are separate videos for northern blotting and southern blotting and western blotting. Please do check it. That's all.